Well, hello anatomy students. Welcome to Biology 201, Human Anatomy and Physiology. Today we're going to be talking about the organization of the body, microscopy, and a lot of our time is going to be spent on histology. Now, at this point in the semester, you've probably already met your lab TAs, you've gone over the syllabus, you should have a really good understanding as far as what's expected of you when it comes to assignments, grades, notebook drawings, and all of the requirements that you'll find in that syllabus. If you are just checking in now or you haven't spent some time or you're still a little bit confused, please make sure you talk to your TAs, email them. They are always happy to help you out. So let's start here with anatomical position. You can see here a woman standing there and her body is upright. She's facing forward. Her arms and legs are straight and her palms are facing forward. Her feet are flat on the ground and her eyes are open. Now this is our basic standard anatomical position. It acts as a frame of reference. So the left side is always the left, the right side is always the right, the superior is always superior, and we can communicate with other medical professionals or other researchers, and we can use this standard frame of reference to understand what each of us are talking about. Now, the same is true with these directional terms. So as you can see over here on the left side, we have a ton of terms that are going to help us understand the direction in reference to our anatomical position. So you can see over here we have ventral and dorsal, anterior and posterior, superior and inferior, medial and lateral, proximal, distal, superficial, and deep. Now, what you'll notice is that these are relative terms, so anterior and posterior. So anterior is towards our ventral side or towards the front or our bellies. Now, you can't just say that my stomach is anterior or my chest is anterior. We need to use it in reference to another structure. So the sternum is anterior to the heart. With our posterior, that's towards the back or towards our dorsal side, we can say that the esophagus is posterior to the trachea. So again, we need some kind of reference here. Let's look at two others, superior and inferior. So superior means above and inferior means below. So the heart is superior to the diaphragm and the liver is inferior to the diaphragm. We can't just say the heart is superior. It doesn't make any sense because it's not referencing something else. So make sure you spend some time looking at these terms, make sure you practice writing out these definitions, and then run through your own body and tell me a little bit about your elbow or your wrist or your knee. When we talk about proximal and distal, you'll notice that these are in reference to the point of attachment. So this is where we would be using these terms on our appendages. So we can say our elbow is proximal to the wrist. It's in closer proximity to the point of attachment or origin, whereas our fingernails are at the distal ends of our fingers. They are farther away from the point of attachment to our bodies. Medial and lateral, I'll just jump back here really quick. Medial is down the center line or towards the median plane, and so our heart is medial to the lungs, and lateral is off to the side. So our eyes are lateral to the nose, or our ears are lateral to the eyes. And then last, superficial and deep. So we're talking about our body surface. So our skin is very superficial, our muscles are deep, but our bones are even deeper. So make sure that you really get a good understanding of these terms. You're going to be using these all semester and in Bio 202 when you get to that point. Now, we can divide the body into different anatomical planes. So as you can see here, we have our subject and she's standing in anatomical position. Now we can divide her body into different planes, and these are basically imaginary surfaces that run through the body. Now our first one is our sagittal plane. Basically our sagittal plane is dividing the body into right and left halves. You can see here, let me back up, you can see here that this line is the mid-sagittal plane. Now, there's only one mid-sagittal plane, and that divides the body into equal left and right halves. We'll talk about parasagittal here in a second, but parasagittal planes divide the body into unequal left and right halves. So maybe you would see a plane running through her shoulder. Our frontal, also called our coronal plane, divides our bodies into anterior and posterior positions. So we can see this purple frontal plane here. And then our transverse plane, this orange one, is going to divide our body into superior and inferior portions. Now here's that mid-sagittal and parasagittal planes that I was talking about earlier. We can see again our subject is in standard anatomical position, and we have one mid-sagittal plane that's going to divide this person into left and right halves. Now you can see the orange lines here are the parasagittal planes, where we have unequal left and right portions. And so we can have infinite numbers of these. We can keep dividing the body into these parasagittal planes. So take a few minutes, run through those anatomical positions, run through those anatomical planes, and when you're ready, come back to the video and we'll move on with some microscopy. All right, welcome back. So at this point in your academic careers, you've probably spent some time looking through microscopes. So a lot of this will be review, and we're not going to spend a whole lot of time on this. But we wanted to just make sure that you are taking care of the microscopes, and you're handling them safely and carefully. We want to make sure you know where to put your specimen on the stage. And we want to just point out the coarse and fine adjustment knobs, because this sometimes gets reversed with students using these microscopes. And then last, just our proper care of slides. Here is our microscope. We've got our ocular lenses up here, our revolving nose piece right down here. And again, our microscopes are a little bit different than what you see in this image. But on that revolving nose piece, we have our different objectives. So we have our low, medium, and high power. Some of the microscopes you'll see have the oil immersion lenses. You will not be using oil immersion lenses in anatomy. We do not use that at all, so you will never go to that objective lens. Now, our low power is 4x. Our medium power is 10, and our high power is 40. And remember, our ocular lenses are also 10x. So you'll be multiplying our low power at 4 times 10, so our low power is going to be 40. Our medium power is 10 times 10, which will be 100, and our high power is 40 times 10, so 400. Again, if you are recording a drawing in your notebooks, in your lab manuals, and you put down 1,000, that is not correct. You shouldn't be looking at that oil immersion lens. You can see this image here has a slide that's put down on the stage. It has some clips, the stage clips that are holding it in place. And then we have our condenser and our illuminator, our light source down here, the power switch, which is going to turn that on. And then we can increase or decrease the brightness of the light with that dial. Again, you have an adjustment on the condenser that will allow you to let in more or less light, and that's going to maybe help you view those slides a little bit differently. Sometimes you might not have a really good view of it. If your light is really bright in there, turn it down a little bit. You might get a little bit more contrast. Over here on the right, you will see that there is the coarse objective knob. That is the biggest one. And the smaller one is that fine focused adjustment knob. Remember, when you're looking under low power, you're going to adjust that coarse focus knob until you bring that specimen into focus, and then you'll fine tune it with the fine focus. From there, You'll turn the revolving nose piece into medium power. You'll find your fine focus, and you'll bring your specimen into view. You will not use that coarse objective or that coarse focus knob when you're looking at medium power. Same is true of your high power. You're going to turn up to high power, and you'll only use that fine focus, because if you turn that coarse adjustment, it moves that stage up, and you can break the slide and run it into that high power lens. When you're moving through our microscopy lab and you're practicing with the microscopes, make sure you're reading and following directions. Remember our field of view and the different amount of light as you are going from low power to high power. Your field of view is getting smaller, and the amount of light that's coming through is also getting smaller. Okay. Make sure you go over your microscope parts checklist. Make sure you know how to focus the microscope, work with the lighting, and again, when you're in your labs, make sure you're taking care to clean the scope. Don't forget, your microscope is like a little baby, and so you're going to have one arm on the arm and one hand on the base, and you're going to carry it and care for it. You're not going to slide it around the table. You're going to set it down gently, leave it there, and you and your lab partner can look back and forth through the scope. But please don't slide that around, and always replace the microscope cover. And when you're done, put it back in the case in the back of the room. Let's spend the last good bit of our lecture today looking over histology. Over here on the right side of your screen, you can see all these different types of images. So these are all different types of tissues, and by the end of today's lecture, you should have a pretty good understanding of how to identify this tissue. So histology is the study of tissues. And what are tissues? Well, if you remember, tissues are multiple types of cells working together, and the extracellular material. They have a particular function, and two or more tissues make up organs, and organs make up organ systems, and organ systems make up an organism. So we have moved from cells into tissues. Now, there are 4 main tissue types that we're going to be looking at, and I would recommend printing out this slide or taking a screenshot of this image and printing this out, putting it in your notebooks, or keeping this thing handy. It's a really good reference tool. You can see up here we've got tissue, and tissue is divided into our epithelial tissue, our connective tissue, our muscle tissue, and our nervous tissue. In 201, we're going to spend a significant amount of time in labs looking at our epithelial tissue, as well as the majority of our connective tissue. We'll be spending a lot of time looking at the fibrous connective tissue, as well as cartilage. The rest of these tissues will be going into more depth in 202. Now, epithelial tissue are one or more layers of really, really closely adhering cells. They are very, very cellular, and they have little to no extracellular matrix. Extracellular matrix is sort of the space and the fluid around the cells. If you think of a brick wall, epithelial tissue would be like a wall made up of a bunch of bricks and almost no mortar in between those bricks. They cover or line parts of the body, and so we can see them covering the external or our more superficial part of our bodies, or they're going to line a body cavity or a tubule. There's no room for blood vessels or nerves to penetrate through this because they are highly cellular and they have very low matrix. And we'll be talking a lot about that when we get into the integumentary system. We also need to make note of this structure down here called the basement membrane. Our basal surface of our cells, which is this bottom part of these cells, is attaching to this non-cellular basement membrane. So you can think of BAS, basal, and BAS, basement. This is kind of like a double-sided sticky tape, if you will. It is a non-cellular adhesive proteins and collagen that helps to anchor our epithelial tissue to the underlying connective tissue. Over on the right, we can see this distinct difference between the cells on the left and the cells on the right. And I'm tracing over where our basement membrane would be. And to the left, we can see our epithelial tissue, and to the right, we can see our connective tissue. Now, epithelial tissue is classified by the cell shape. So we can see that we're going to have flat, squished cells called squamous. We have cube-shaped cells called cuboidal. And we have tall, columnar-shaped cells called columnar cells. They're also classified by the number of layers. And so if we have one layer of cells, we call this simple. If we have multiple layers of cells, we call this stratified. Looking at the differences between simple epithelial tissue has one layer of cells which are connected to that basement membrane. And so we see one layer of cells here. So we know this is simple, and then we're going to look at the shape of those cells. So we might look at these and say, okay, they are columnar shaped, so this would be a simple columnar epithelial tissue. If we look at a stratified epithelial tissue, we're first going to find where that basement membrane is, and we're going to find our basal cells, or the cells connected to the basement membrane. And we're going to see if there are other layers or multiple layers. And in this example, we see we've got 3 layers. But the difference is that we are going to name the cells by their shape based on the shape of these apical cells right here. So this top layer is called the apical cells. This bottom layer are the basal cells, and the basal cells are going to adhere to the basement membrane. So here, if we look at the cell shape, we've got a simple cuboidal, and here what we would say is that we have a stratified cuboidal tissue. Keep in mind, when you're looking through a microscope, these are really, really thin slices of tissue that you're looking at that have been prepared on these slides. So as we have in this image an egg, depending on where the cross-section goes through this egg is going to give us a different perspective of that egg. So if we look at our mid-sagittal cross-section, we can see we have the largest view of that egg and its yolk, or its nucleus. If we look at the green that are these parasagittal and a little bit lateral to that mid-sagittal line, we can see that the egg looks a little bit smaller, or the cell looks a little smaller, as does that nucleus. But if we look at these blue cross-sections, these parasagittal cross-sections almost completely laterally, we see a very small circle relative to our mid-sagittal line, and we see no nucleus or no yolk in this egg. So again, we know that that egg has a yolk. In our cross-section here, we just don't see it. Now the same is true when we're looking at different cross-sections. So here on the left, we can see a macaroni noodle. Depending on where we take that cross-section, right here at this top or most superficial dotted line, this red dotted line, if we look down here on a microscope, we might see these 2 circles, and we might look at them and say, oh, well, there's a tubule, but we don't actually know if they're connected. So what we would need to be doing is looking at many, many, many slides that are very they would go deeper and deeper and deeper through this macaroni. So here at the green line, right in the middle of our macaroni, we can see that we have these circles that are kind of connected. And then if we look down here at our blue dotted line, we get sort of this oval shape. So 3 different views of the same noodle, and they all give us a little bit of different information, so we can sort of put all these images together. Over here we have a squiggly sweat gland, and if we take a longitudinal section, we're cutting down through it, again you can see we see different parts of that sweat gland, but we don't see the entire sweat gland. And then last, we have a few different different sectionings that you'll see on your microscopes. And so you might see the abbreviation LS or CS or XS, TS. These are going to give you an idea of what type of section you're actually looking at. So an LS here would be a longitudinal section. Again, it's going to give you a different view of that structure that you're looking at. We might see a cross section or a transverse section here, again giving us a different view of what that tube looks like. And then an oblique section where that tissue is actually cut at an angle, and again that's going to give us a slightly different view of what we're looking at. Let's jump into some tissues here. Now, take a few seconds, look at this tissue and tell me or think about what it is that you are looking at, and think about how we identify epithelial tissues. So do you see layers, and do you see any specific shapes of cells? So let's look at this a little bit. We can see here we've got some cells lining this surface here. We see sort of maybe an empty space here, but remember, epithelial tissue is lining an external part of our body or a cavity or a space, and so we do see something in here. It could maybe be some residual dirt, or it could be something inside of a tubule, and so right now we wouldn't expect you to know exactly what that is, but we would be kind of expecting that you would be able to figure this out down the road. But right now what we want you to be able to do is identify what kind of epithelial tissue we'd be looking at. So if we look at these cells, we see that there is one layer, and they are slightly flat and squishy. They're definitely not columnar shaped, and they are not cuboidal. They're very rounded to squished. So this is in fact our simple squamous epithelial tissue. This tissue functions for rapid diffusion, and so gases can pass through this plasma membrane, through this flat squishy cell, and out to the other side. We might see nutrients passing through here, and so it creates a smooth lining, and it also secretes serous fluid in different tissues. Where we'll find this are in our lungs, inside the heart, and inside of our blood vessels, just as a few examples here. Now, for tests, weekly tests, you're going to need to be able to identify this tissue based off of its appearance. You're going to need to give us a function of this tissue, and you're going to need to be able to identify a location. So spend time looking through your microscopes. Really look at this tissue. Start on low power. Look around. What does it look like? Where you might look through a microscope on that slide, you might spend just a few minutes. You'll take a photo with your phone, and then you'll move on. You'll go home, you'll draw it, and you're like, yeah, I got this. But then you show up, and your TA has a test question set up on a different slide in a different part of that slide, and it doesn't look familiar to you at all. So explore the slides, use that low power, scan around, move into medium power, scan around, really get familiar with it, move into high power, what does it look like, and back out. So really get familiar with this tissue, spend time coming to open labs. A lot of this tissue is new for many of you, and this is one of the biggest challenges of this lab, is understanding tissues. It doesn't go away. Pretty much every lab in 201, you're going to be looking at different tissues, and in 202, every lab you're looking at different tissues. So this is really foundational information. Okay, take a second, look at these cells and think about what it is you see and what kind of tissue it is. All right, so hopefully what popped out to you was this single line of cells on top and here on bottom. And we also see a circle right in here, and we see almost a complete circle over here in the top right. Now, again, in both of these images, we see that there is a single layer, and if we look at these individual cells, they look fairly cube-like. They definitely don't look flat and squishy. So this is our cube-shaped cells that are lined up in a single row, which means that we are looking at our simple cuboidal epithelial tissue. Now, what does this tissue do? It's involved in absorption and secretion. So these cuboidal cells produce hormones, they make sweat, they make oils, and then they release them through tubules, which is what you're looking at. And we have two different cross sections of some tubules here. Here we have a longitudinal cross section. If you think about this as a garden hose, it would be cut down the lengthwise. If we look at this circle at the bottom, it would be if the garden hose was coming out of your computer monitor at you and you cut through it, you would see these cells would be essentially the rubber, and inside here would be the lumen, or where the water would be traveling. So we find these in kidney tubules, glands, the liver, just to, again, to name a few locations. On tests, you'll likely only need to know one, unless your TA specifies that you should know more than one function or location for those tissues. All right, go ahead and pause your video here, take a few seconds, and think about what it is you're looking at. All right, how'd it go? So down here, let's identify our most important layer here, or our most important structure is our basement membrane, a dark staining double-sided sticky tape adhering to the basal cells, or the basal portion of our cells. Now we notice that we have a single nuclei in these cells, and we have tall columnar shaped cells here. So we do have a single row of these narrow cells. They're vertically oriented, and their nuclei tend to be in the basal half of the cell. So this is in fact our simple columnar epithelial tissue. What you'll also notice with this tissue is these big goblet cells, and when you're looking through your microscope slides, you'll see them sometimes as clear and transparent, but they are associated with our simple columnar epithelial tissue. They're actually single-celled glands that secrete mucus. That mucus helps lubricate and trap different things in that mucus. Now you'll also notice up here in this little brush border here, these are called microvilli. They are extensions of the plasma membrane, and those extensions of the plasma membrane increase the amount of surface area. And so these microvilli help with absorption. So we tend to find this in our stomach through our intestines, where we need lots of surface area to absorb as many nutrients as possible. Okay, go ahead and pause your video here and think about our last slide in comparison to this slide. All right, how did you do here? So let's look at a couple of these structures. Here's our basement membrane at the bottom again. We see these nuclei in those cells, but we also see these other nuclei here. Now these are nuclei of basal cells. And these basal cells make this tissue look stratified. But in fact, if we look at this cell right here, it looks like it doesn't, it looks like it stops right here. But in fact, this cell actually goes behind this basal cell and comes in contact with that basement membrane. So we have this stratified appearance, but when in fact it actually is not. So we call this pseudo-stratified or falsely stratified. So pseudo means false. One of the other things that you'll notice here is that we have our goblet cells again, so a similarity with our simple columnar epithelial tissue. So again, this is going to secrete mucus, and it's going to help trap dust particles. At the top, or the apical portion of these cells, we see cilia. So these little hair-like projections that beat and wave, and as dust and particulates get trapped in the mucus, these cilia help to move those particulates away from the inside of our lungs. And so our cilia are helping to move or transport substances, where our microvilli are helping to increase absorption. All right, next let's look at some stratified epithelial tissue. Now remember, there is more than one layer of cells, and they are again named for the shape of those apical cells. The one exception is transitional epithelial tissue, and we'll look at that here soon. Now there's two variations that you'll see: keratinized epithelial tissue, which actually has a layer of dead cells, and non-keratinized epithelial tissue that does not have that layer of dead cells. So if we first look at this image, there's a lot going on here, and one of the problems with looking at histology on a PowerPoint through an online recording on YouTube is that you lose a lot of the detail of some of those plasma membranes. So again, this is why it's really important that you make time to get into lab and open labs and spend time looking at these tissues. You probably noticed a few things here. You probably saw this dark line going right here through the middle, but don't let that confuse you. This is actually not our basement membrane. You'll learn more about this layer in lab 2. Our basement membrane is down here where this dark purple stained tissue or dark purple stained cells meets this lighter staining tissue down here. And one of those differences that you'll notice is that we have lots of dark staining nuclei, these dark purple dots. If we look into this tissue, we see that those dark staining dots are scattered throughout. And this is in fact actually a connective tissue, and we're going to talk about how you identify that here soon. When we talk about this slide here, we can see that our basement membrane is down here. We have these nuclei, and this entire layer is actually our multi-layered epithelial tissue. From this black line all the way to the top where we see just white, this is again indicating that there's either the external environment or a lumen or some kind of cavity. But we see that these cells up here are actually dead. We don't see any nuclei in these cells up on top. And so we have this really thick layer with these dead squamous cells, and they're packed with protein, with the protein keratin. So that makes them really hard. And so that layer helps prevent penetration by organisms. It reduces abrasion, prevents water from leaving through our skin here. So we find this in the epidermis, and again, when we talk about our integumentary system, you're going to learn about all the different layers that make up this image that you're seeing right now. This is our keratinized, stratified squamous epithelial tissue. Now, if we look at this image, here you can see our epithelial layer, and here in dark purple we see this basement membrane. We see our basal cells here, and those cells change shape And when we look up here, we see these flat squished cells, which indicates that they are squamous cells. Now, one of the other things to note is that you can see we have some nuclei that are in here, and we also see some cells that don't have nuclei. It can be tricky to say, oh, well, these must be dead and this must be keratinized, but because you're seeing the nuclei throughout this multiple layers, it's a good indicator that that tissue is likely alive. It's just not getting cut through on this image. So this is our non-keratinized stratified squamous epithelial tissue. It lacks that surface layer, but it creates a moist, slippery, abrasion-resistant layer. And so we find this on our tongue, in the oral mucosa, the esophagus, and the vagina. Pause your video here and think about all the stuff that we've gone over, and what are some of the differences that you notice here. So hopefully what you noticed here was that this is multi-layered, and so we have a difference in staining right along here. It's kind of hard to see that direct basement membrane, but again, much more clustered. This tissue up here is much more clustered with nuclei. Down here, it's much more sparse. Those nuclei are more sparse. So at the top, what's different though is we see these circular, rounded apical cells. I like to think of these as blobs of frog eggs. One of our TAs also said caviar on crackers, and so you can think of the cracker as that basement membrane and the caviar or the fish eggs sitting on top of it. So this is our transitional epithelial tissue. This is one of the hardest tissues to identify in a microscope. You might look at this and be like, oh, okay, I totally got that. A lot of students show up feeling pretty confident about their histology, and they see this one and they tend to call this stratified squamous epithelial tissue on a test, or stratified cuboidal. So please do make sure you spend some extra time looking at our transitional epithelial tissue. What this tissue does, these round cells actually stretch to allow for filling in that bladder or along our urinary tract. And our urinary tract kind of runs from our kidney through our ureter and into our bladder. And so those rounded cells actually flatten out when it's stretching, and after we relieve ourselves and eliminate that fluid or that urine that's in our bladder, those cells will recoil a little bit and become more rounded. Now, I put this in your PowerPoint here as a review. I would spend some time printing this out and reviewing this, going over the location and the function, and practicing getting a good understanding of how we name these different tissues. Take a few minute break, digest epithelial tissue. We'll come back here in a few minutes and pick up with connective tissue. All right, welcome back. Let's pick up with connective tissue. Now, connective tissue has widely spaced cells that are going to be separated by fibers in that matrix. So back to our brick wall reference. With epithelial tissue, we said there's lots of cells and almost no mortar. With connective tissue, we would have a wall made of lots of mortar and very few bricks. So here we can see that there is more matrix than cells, where in epithelial tissue, it's much more cells, or many more cells, than the matrix. So that's going to be your biggest indicator when you're looking through your slides and practicing in labs. This is the most abundant tissue and the most variable tissue. We're going to see a lot of differences in that tissue structure. So there are different types that we're going to need to learn about. Now, we're going to be focusing on fibrous connective tissue, also called connective tissue proper. In fibrous connective tissue, or connective tissue proper, there are two main types. The first type is called dense connective tissue. So in dense connective tissue, we see that the fibers, or there are lots of fibers that are filling the spaces between the cells. So back to our brick wall. In order to identify epithelial versus connective, we say, is there more matrix than cells? If so, we're talking about connective tissue. Now, when we look at our fibrous connective tissue, we say, do we see more fibers than cells, or do we see more cells than fibers? So a slight nuance there. With dense connective tissue, we're going to see more fibers than we do cells. And we have three types of dense connective tissue: dense regular connective tissue, dense irregular connective tissue, and elastic tissue. The other type is loose connective tissue, if you didn't see that one coming. This is a gel-like substance, fills the spaces between the cells, and here we're going to see more cells than we do fibers. So there's three types: reticular, areolar, and adipose tissue that we'll be looking at. Now, we're also going to learn about cartilage this semester. So there's three types of cartilage you'll be learning about: hyaline, fibrocartilage, and elastic tissue. Elastic cartilage. You're going to learn these different cartilages in your articulations lab. That articulations lab is open and it is fully online. The lecture is online. All of the slides will be you'll be viewing in labs or in open labs on your own. The test is online. You're not doing the articulations lab in your actual labs per se. Okay, so make sure you talk to your TAs about it. They will be announcing and giving you updates about that. Check your syllabus. You have multiple weeks to complete this, so I wouldn't wait until the very end. Try to piece it out. Watch the lecture 1 week, maybe the next week go into lab or open lab and start looking at some of those slides. And then the week after, take your test, turn in your assignments. The other type of connective tissue we have is bone, which you'll be learning a lot about next week and in your lectures. And then we have blood. This is our liquid connective tissue, and you'll be learning a lot about blood in 202. So pause the video here, take a moment, and think about what it is that you see here. How is this different from what we learned about with epithelial tissue, and how can you get your bearings straight? What we're looking at here are these densely packed, parallel, wavy collagen fibers. So this is our dense, regular connective tissue. Lots of fibers, and it is regular, meaning it is oriented in a specific direction. So the function of dense regular connective tissue is that it holds bones together or holds muscles to bones. And so we find this in ligaments and tendons. Now, on the right and left sides, you'll notice that the fibers are running from right to left. So this is really strong and is going to withstand tension from a left and right direction. If there was a force applied to this tissue from the top or from the bottom, this tissue is not as strong. And so when you think about football or soccer injuries where someone tears one of their ligaments, an ACL or an MCL, and they've been impacted from a lateral direction, the forces are hitting this tissue in this direction, and it's a little bit weaker as opposed to tensions coming in from the left or the right. Looking here, this is much different than what we just saw. And so in this image, we see collagen fibers, but these collagen fibers are going in random directions. Some are coming out of the screen at you. Some are going to the right and to the left. Some are going to the top and the bottom. Some are going at different angles. And so this is our dense irregular. There's no regularity of those collagen fibers. So this is going to withstand stresses that are applied in all directions. So if there was a force coming out of the screen, if it was going to the right and to the left, from the top and the bottom, it's going to be able to resist those stresses in all those directions. We find this in our dermis, we find them around cartilage, we find them around bone. Next image, take a few seconds here and see what you notice that's maybe different in the last two. All right, one of the things that should be sticking out to you are these dark squiggly lines. This is the protein elastin, and this you'll also be able to identify in labs some nuclei in there from the cells called fibroblasts that are going to produce that elastin. So this is our elastic tissue, and so the protein elastin is stretchy. In this example here, we're looking at an artery. Okay, next, when we look at this image, one of the things we should be noticing is that there is a really loose network of fibers and cells, but in this image, we're seeing lots more cells than we did in the other three images, and a little bit less fibers. Here we're looking at loose connective tissue, and this is our first type of loose connective tissue, our reticular tissue. So it forms a framework or a supportive stroma for different organs. Some of those organs are the lymph nodes, the spleen, and the thymus. You'll learn a lot about the immune system or the lymph system when we get into 202. All right, our next image, we've got a few different fibers identified here. You can see collagen, elastic, and reticular. We can see some dark staining cells that are in this image as well. And so again, a very loose arrangement of these fibers and cell types. And so lots of ground substance or lots of matrix here. So this is our areolar tissue. We find this under our epithelial tissue in the dermis. It forms passageways for nerves and blood vessels. And when we look at this image, this is going to allow these blood vessels and nerves to move through that matrix and through these fibers. When you think about epithelial tissue, all those cells are really tightly adhering to each other. There's no room for these blood vessels to move through. So it's relying on our underlying connective tissue for nutrients to diffuse up and wastes to diffuse out. So again, we can find this around many organs. We can find this between our epithelial and connecting from our epidermis to our dermis. All right, and then when we look at our last slide here, you might be looking at this and saying, okay, well, I see some plasma membranes. This is very thin, and don't let it fool you into thinking that this is like a simple squamous, but we have these really big empty spaces here. And so it's almost like you can think of these as like inflated balloons. So these plasma membranes out here would be the rubber, and what you'll notice is you do see some cells, but those nuclei of those cells have been pushed out to the side, and so you can kind of think of those as a little, nozzle or the knot of the balloon. And so they're very empty, and this is going to allow for fat storage. So this is our adipose tissue. We can store energy. It also is really useful for insulation and cushioning around different organs. So we can find it in the fat below our skin, breast tissue, surrounding different organs to help, again, prevent any damage to those organs. So we've made it. We've gone through epithelial tissue. We've gone through our fibrous connective tissue. So now your goal is to practice, practice, and practice. Remember, this is foundational material for 201 and 202. Please do not take these tissues lightly. Spend a lot of time up front. Spend extra time going through it. Print these images out of this PowerPoint. Put them into plastic sleeve covers. Label them, draw on them, pull one out, erase it, put another image in. Do this over and over again until you really have this down. Spend as much time this week as you can in open labs practicing this. Use your histology DVDs to make yourself quizzes. You can even use this PowerPoint as your own quiz. You can just take the slides and delete the text off of it and run through the slides practicing it. When you get into labs this week, spend a lot of time looking through those microscopes. Too often now with smartphones, people come in, they look at a microscope, they take a picture of it, and they're like, I gotta go, I'm gonna get out of here. You've already scheduled your time in lab. Spend that time looking through the microscopes. Talk about it with your lab mates, and take turns throwing a slide onto a microscope and say, hey, what do you see in here? Can you describe it? Tell me what it is. And then have the person do it to you, and just go back and forth through that. So thanks for tuning in. Good luck this week working with histology, and we'll see you next week when we go over the integumentary system.